All right, good morning, everyone, and I'm excited to introduce our speaker today, Dr. Ben Richardson, Assistant Professor of Pharmacology at Southern Illinois University School of Medicine. Dr. Richardson is a distinguished neuroscientist and neuropharmacologist whose research delves into the intricate workings of brain circuits involved in sensorimotor behaviors, particularly how these circuits are impacted by environmental and genetic factors in neurodevelopmental disorders like autism spectrum disorder and ADHD. His innovative approach combines electrophysiology, viral manipulations, optogenetics, and behavioral analysis to uncover the synaptic and circuit changes underlying these conditions. In today's webinar, Dr. Richardson will discuss the disruption of brain regions often overlooked in neurodevelopmental disorders, explore the interaction between sex and stress, exposure on cognitive and affective behaviors, and examine the synaptic changes in the cerebellum that may drive behavioral alterations in autism spectrum disorder. Please join me in welcoming Dr. Ben Richardson.
Hi, Chloe. Thank you. It's great to be here and be part of the Precisionary Instruments webinar series. Our lab has used the compressed that's produced by Precisionary for a lot of our work. And that's what I want to focus on talking about today is how we use ex vivo brain slices, again, prepared with a compressed to try to identify some of these specific mechanistic changes that we see in a few different animal models of neurodevelopmental disorders. And so one of them being an animal model of autism spectrum disorder, and another one is an animal model that I'll sort of introduce as new. Our lab has been developing this as a way to assess the impact of early life stress or early life adversity on circuit function and cognitive and affective behavior. So before I go too much further, I just want to say that I don't have any financial conflicts of interest to declare. And you sort of laid out already, Chloe, some of the major learning objectives that I hope to hit today, but I do want to give those as sort of a guide for exactly what I'll be talking about. So to be clear, I hope one of the things that folks come away from today's session with is an appreciation of maybe how to consider different brain areas that we don't always think about as sort of those canonical or typical brain areas that we normally would think about in terms of areas that drive changes in cognitive behavior and affective behavior, but thinking about a few different ones that might also participate and thinking about them based on their sensitivity to disruption in response to factors that are linked to different neurodevelopmental disorders like autism, ADHD. I'm going to talk about two projects today. The first one I'll talk about is led by a PhD student in the lab, Raj Chetri. So Raj is trying to identify specific synaptic changes that are occurring in the cerebellum of an animal model of autism spectrum disorder. And then the second project I'll talk about today is a little bit newer, and it's led by another PhD student in the lab, Savannah Brannon, and she's developed a model of early life stress where we see specific changes in the activity of neurons in the locus coeruleus, and in parallel, changes in cognitive and affective behavior in a specific mouse model. So I'll start talking about our project related to autism spectrum disorder, ASD, first. And so many of you may be aware of or have heard about ASD, most likely these days. Each year, the incidence of ASD continues to rise. And right now, I believe the number is expected to be that 1 in 36 children in the United States are affected by autism spectrum disorder. And so ASD is, I would say, characterized by changes across a number of different behaviors. But one of the things that some people say is that if you've met one person with autism, you've really only met one person with autism, because the way ASD manifests in different people is so different from person to person. In terms of the severity of symptoms, it ranges across a spectrum. And in terms of the domains and type of behaviors that are affected, it also ranges from several different types of behavioral domains. And so whether that's changes in executive function, communication ability, sensory processing, the emergence of increased repetitive behaviors, there are all different types of combinations of behavioral changes that occur in autism spectrum disorder. Now, when it comes to the cause of ASD, there are a number of factors and type of factors that have been linked to ASD. So some of those are biological factors like neonatal hypoxia. Others are environmental factors like stress or exposure to pesticides. although the mechanisms by which these types of factors drive the development of ASD are not as well known as the genetic factors that contribute to ASD. And so some of what we know about the genetic causes of ASD come from syndromic ASD when it's present in individuals with other syndromes like those with Fragile X or Phelan-McDermid syndrome. And so I'm going to use what we know about the genetic risk for ASD to talk about how we are beginning to understand a potential role for the cerebellum in autism spectrum disorder. So there have been a number of studies over the last couple of decades that have looked at the genome of individuals with ASD and tried to identify genes that are mutated or disrupted in ASD. And these studies, all in all, have identified anywhere from 100 to 200 genes that are disrupted in individuals with ASD. But most recently, there was a study led by Joe Buxbaum and others where they evaluated the genome of almost 12,000 individuals with ASD. And in doing so, they identified at least 102 genes that were associated with autism spectrum disorder. And when they looked at the expression of those genes, they found that of those 102 genes, many of them were expressed in the cortex of the brain, the cerebral cortex, and a similar amount were expressed in the cerebellar hemisphere or the cerebellar cortex. Now, when they dug down a little bit further, they found that 53 of those 102 were associated with, predominantly associated with ASD. And it actually turns out that there was a higher expression of those 53 ASD predominant genes in the cerebellum than there was even in the cerebral cortex. Now, this relationship between expression of ASD-linked genes in the cerebellum is something that has been around for a while, but maybe hasn't been able to be probed at that level. Going back a decade or so, there are some meta-analyses that looked at the expression of different ASD-linked cohorts of genes. And so they also found that if you evaluate the expression of those cohorts of genes, that there are many more of them expressed again in the rodent cerebellum. So this is a mouse cerebellum where we're looking at the normalized expression of different combinations of ASD-linked genes. And we see there's a much higher level of the expression of these genes in the mouse cerebellum relative to other parts of the mouse brain. And the last thing I'll say about what we know about, although there is a lot more that we could say, the last thing I'll say that we know about the relationship between the cerebellum and ASD is that if you look back at a meta-analysis from about 10 years ago, Sam Wong at Princeton University led a review study, sort of a meta-analysis, and they identified that the single highest non-heritable ASD risk factor was actually cerebellar damage at birth. And when they considered all risk factors, this cerebellar damage at birth was really only the second highest cause or identified risk factor for autism spectrum disorder, next to only having an identical twin with autism. So these data and others that I don't have time to talk about today have begun to develop this link between what happens in the cerebellum in response to these factors that are linked to autism, and with that, opening up questions about how these changes change cerebellar function and how might they actually contribute to changes in behavior akin to the disruptive behaviors we see in ASD in humans. And so before I go too much further, not everybody is always up to date on their cerebellar anatomy and function. So I just want to cover that real quickly here. So when we think about the cerebellum or cerebellar structure, the cerebellum is this region that sits in the back of the brain just below the occipital cortex. And it has two main components. The first major component is the cerebellar cortex. And that's what makes up a bulk of the mass of the cerebellum. And the computations that happen in the cortex, and I'll talk more about the circuitry of the cerebellar cortex here in a little bit, but what happens in the cerebellar cortex in large part determines the activity of neurons in the cerebellar nuclei. There are 3 cerebellar nuclei on the left and 3 on the right, so 6 total. And these 3 nuclei classically are known to project out to a number of different motor brain areas. And through these projections, they shape the activity of those motor brain areas. And very simply, what the cerebellum is doing here is taking input from the outside world and input about what we expect to happen in the case of a motor command. It's making some comparisons about what's happening and what we expect. And then based on these computations that occur primarily in the cortex, it shapes the activity of these motor brain areas to regulate things like balance, motor coordination, and motor learning. And so one of the ways to, I think, simply think about what the cerebellum is doing is that it uses this predicted outcome and compares it to what's actually happening so that we can appropriately alter our motor behavior or motor commands in real time. So one way to visualize how this prediction is used is in this example here. If you have an individual who is asked to move their finger from a point in space to their nose, a person with a normal cerebellar function would do it quite efficiently and probably in a straight line. But someone who has some cerebellar damage that affects the motor system, motor aspects of the areas of the cerebellum, they may do that a bit more inefficiently and might move through a more wandering path, but eventually they would get to the intended spot. And one maybe overly simplistic way to think about what the cerebellum is doing is in some ways it's sort of behaving like a lane keep assist on a car. So it's sort of keeping us between we're within the margins of error so that we can achieve the task that we intend to achieve. Now, when you look at this figure that I showed earlier about the different types of processes and functions that are abnormal or disrupted in ASD, you kind of wonder why we're focused so much on a structure like the cerebellum that is strongly associated with motor processing and motor function. Well, if you look at all of these different types of behaviors, it does turn out that one of the major areas in which individuals with ASD have challenges is in their motor skills and motor function. Whether it's gait or more fine motor processes, there often is some deficit in these areas in individuals with ASD, but that's not really all there is to it. So again, classically, we do think about the cerebellum and its role in the motor system. But more recently, we've begun to identify and understand how the cerebellum is projecting to, and that is the output nuclei of the cerebellum, is projecting to and interacting with a number of different brain areas that are not necessarily associated with motor function. And so at a minimum, we know that the output of these cerebellar nuclei is projecting to many areas of the thalamus, both sensory, limbic, and motor thalamic areas. But more than that, a number of other studies have gone on to show not only is the cerebellum projecting to non-motor brain areas, but these projections are actually capable of modulating different types of behaviors that, again, are not specifically motor behaviors, whether that's spatial learning through its modulation of hippocampus, social and repetitive behaviors through modulation of medial prefrontal cortical areas, reward and social engagement via the VTA, anxiety through the periaqueductal gray. And this is just a subset of what we know about how projections out of the cerebellum are engaging in and modulating processing that's occurring in parts of the brain that are not part of the motor system typically. And then in people, others have been trying to develop a map of areas across the cerebellar cortex using fMRI to identify areas where activity is increased during different types of processes and activities. And there are areas that are regularly activated during emotional processes, spatial orientation, language, executive function, and so on. And right now, one version of that map sort of looks something like this, where we see collections of different hotspots or areas of activity when individuals are engaged in different types of motor, but also non-motor tasks. And so our lab has kind of taken that information and used that to drive some of our interest in the role of the cerebellum and specifically granule cells in the cerebellum that I'll talk about today. And we take what we know about the fact that there's this increase in ASD-linked gene expression in the cerebellum, especially in the cerebellar cortex. And we now know that through a lot of emerging work, this is a very active area of research, that there is some degree of cerebellar modulation of multiple non-motor areas and their related processing. And so with that, we have a couple of very simplistic, I think, basic questions that start with how do ASD-linked genetic factors primarily affect cerebellar cellular and circuit function? And we could go with non-genetic factors, but I think because of this relationship between so many clear genetic risk factors, potential genetic causes of ASD, focusing on some of these genetic factors has been a way to use to leverage animal models in a way that gives us a lot of specificity and a lot of power. And then the second question we ask is, does ASD, like behavioral phenotype, relate to cerebellar dysfunction in any way? That one is a little bit more challenging to address depending on what part of the cerebellum you're working in, but that's something that we're also actively engaged in. So as we started this work on trying to understand the role of the cerebellum in ASD, we referenced the genomic data that were out there. And one gene that reliably pops up as being a risk factor, mutations or deletions in this gene being a risk factor for ASD, is the SHANK3 gene. And actually, anyone with a mutation or deletion in the SHANK3 gene is typically diagnosed with something called Phelan-McDermid syndrome. And somewhere around 95 to 99% of individuals with Phelan-McDermid syndrome, or PMS, also have a diagnosis of autism spectrum disorder. And part of why we chose SHANK3 was because of its high level of expression in the cerebellum. So this is in situ hybridization, looking at mRNA expression of SHANK3 across the cortex, the striatum. You can see some expression in the hippocampus. But the interesting thing for us was there's a relatively or rather high level of SHANK3 expression in cerebellar granule cells. And it looks exceptionally high because these cells are so dense. Cerebellar granule cells are in number some of the more numerous cells of the brain. And in fact, there are more granule cells in the cerebellum than there are other neurons in the rest of the brain. And so as we move forward here, let's just talk for a second about SHANK3 function, what it does and where it's expressed. So SHANK3 is this multi-domain protein that sits at postsynaptic densities. And this is data that we know from a number of different other functional studies that has evaluated the role of SHANK3 in other brain areas outside of the cerebellum, like the striatum or the cortex. And as SHANK3 sits here in these postsynaptic sites, it's actually working to work as a scaffolding protein to help regulate and modulate the expression and membrane insertion of glutamate receptors, specifically NMDA, AMPA, and metabotropic glutamate receptors. Now, the SHANK3 gene has 22 exons, and those exons are differentially spliced to reliably produce 6 different protein isoforms, A through F. And if you were to look at the expression of SHANK3 using a Western blot, and you used a sample from the entire brain without the cerebellum, you would see that there are these different bands of SHANK3 protein corresponding to the different isoforms. In this case, we're just seeing A through E. And then if you were to do that same Western blot with tissue only from the cerebellum, you would see that the cerebellar specific isoforms are limited to isoforms C and D. And so now we have data that suggests that there's a specific expression of isoforms C and D of SHANK3 that appears to be largely expressed in cerebellar granule cells. And so as we now develop our approach to trying to understand the role of SHANK3 in the cerebellum, we decided to use a mouse model that was developed by Joe Buxbaum's lab. And this mouse is lacking exons 4 through 22 of the SHANK3 gene. So this is a full-length SHANK3 gene and all of its exons. And the SHANK3 4 through 22 knockout mouse only has exons 1 through 3 remaining. And if you look at the expression of different isoforms, we see that in a wild-type mouse, using a Western blot, you see many of the different isoforms expressed in the brain. But in this knockout mouse, essentially all of those isoforms are completely gone. Now, when our lab started to use these SHANK3 exon 4 through 22 knockout mice, one of the first things we did was start to do some behavior with these mice. And being a cerebellar lab, we first started to evaluate their behavior on the rotarod. So this is a rotating rod where mice are put on the rod and asked to stay on it. And naturally, they will try to stay on it before landing to a landing area. And the maximal time they can stay on is 5 minutes or 300 seconds. And we started by evaluating the behavior of juvenile mice, ages 5 to 7 weeks. And we were a little surprised that we didn't see a larger behavioral deficit in the rotarod. So here you see the time to fall on the rotarod for wild type juvenile mice in black and knockout juvenile mice in gray. And yes, there is a reduction in their time to fall in terms of the reduction that is for the knockout mice, but this change was rather small, we thought. And so it was especially next to some of the published behavior that had been done by the Bucksbaum lab using these mice. And so we evaluated other aspects of these mice that had been determined in the literature and noticed that in older mice, people were seeing much larger deficits. And so we decided to do a full age-dependent study to evaluate the behavior of these SHANK3 mice. And when we tested the adult SHANK3 mice, SHANK3 knockout mice, now we see a much more robust reduction in the time to fall in the knockout mice relative to the wild type mice. So knockout mice here would be in light pink, and the wild type mice here would be in dark red. And we performed a number of different behavioral assays with these mice at 2 different ages to try to more fully understand the age-dependent development of some of the behavioral phenotype that had been observed in these SHANK3 knockout mice. And so as we evaluated their behavior in the open field, where we looked at the open field center time of these mice with reduced open field center time corresponding to increased anxiety-like behavior, again, we saw that there was a modest reduction in open field center time in the knockout mice that were juveniles relative to wild type mice. But it was not until we tested the adult cohort when we noticed a much more robust reduction in the open field center time, indicating elevated anxiety-like behavior in these mice. And similarly, we went on to evaluate the social preference of these mice as well. And as juveniles, there really wasn't a significant change in social preference between the SHANK3 knockout and wild type mice. But again, now once we got these mice to an older time point at the adult 3 to 4 month range, we saw a significant reduction in social preference for these mice. And so we had a couple people help out with this work, primarily James Beavers and Raj Chetri, so a master's student, and James, who's now a medical student, and Raj, who was still a PhD student in the lab. They assessed these mice on a number of different behaviors, and these behaviors sort of fell into 2 groups. So some of these behaviors, the genotypic dependent differences were already present in the juvenile group. And that was to some degree something we saw in anxiety-like behavior in certain cases, but then in others, locomotor behavior and avoidance and anxiety behavior like marble burying and grooming, these behaviors were already established in full in juvenile mice. Whereas motor function, sociability, and to a degree some anxiety-like behaviors were not really the differences were not there in full force until mice reached that adult time point. And so now with this in mind and the fact that these specific behaviors that we noticed did not fully manifest until mice were a little bit older, these behaviors specifically have been linked to cerebellar, some dependence on cerebellar processing and function. We asked the question, where is SHANK3 expressed and what is it doing in the cerebellar cortex? And this is where we dive into the anatomical and cellular function of SHANK3 in the cerebellum. And so, again, if we look at the expression of SHANK3 mRNA, and so we see mRNA expression in a number of brain areas, including cerebellar granule cells. Before we go too much further, we should probably talk about the anatomy of the cerebellar cortex. So as it relates to these granule cells, cerebellar granule cells sit in what is called the granule cell layer. If you look at this image, you can hopefully appreciate there is some layering of some lighter intensity, darker intensity, and essentially no intensity that corresponds to the different layers of the cerebellar cortex. And throughout this layering, we have a distribution of different cell types within the cerebellar cortex. And so in the granule cell layer, we have mossy fibers that are glutamatergic fibers coming into the granule cell layer. And these mossy fibers may express either VGLUT1 or VGLUT2, depending on their source, but they will come in and release glutamate onto granule cells that all reside in this granule cell layer. These granule cells then form axons that shoot up through the Purkinje cell layer that houses the cell bodies of Purkinje cells, on up into the molecular layer where they bifurcate and form parallel fibers that then release glutamate onto and increase the excitability of Purkinje cells. And so granule cells at this part, within this location of the circuit, effectively work to integrate all excitatory glutamatergic signals that are coming into the cerebellar cortex, at least at the level of the granule cell layer. And so I had an undergraduate student who was interested in trying to understand what exactly was the nature of this SHANK3 distribution within the cerebellar cortex. And so Romana, again, was an undergraduate at the time and is now a graduate student at Einstein Medical College doing some really interesting work on the role of oxytocin in the cerebellum. I like to hope or like to think that she got her interest in the cerebellum from her work in my lab, where she did the following. It's very simple behavior, but we were pretty surprised at the result and encouraged at the same time. So what she did was to use some immunohistochemistry to stain sagittal sections of the mouse cerebellum for VGLUT1 and VGLUT2 to label the two different types of glutamatergic terminals that enter the granule cell layer. And if you zoom into this area, you can see the distribution of those VGLUT1 in blue and VGLUT2 in magenta terminals that come into the granule cell layer. And then here you can see the cell bodies of the Purkinje cell layer and the molecular layer up here at the top. And as she looked at the expression of VGLUT1 and VGLUT2 relative to SHANK3, she noticed that everywhere she saw VGLUT1, and down here it's in magenta with SHANK3 in green, everywhere she saw VGLUT1, there seemed to be SHANK3 wrapped around that VGLUT1 signaling or co-localized with it. Everywhere she saw VGLUT2, it's pretty much the same thing. When she looked at the co-localization of VGLUT2 and SHANK3, she saw that everywhere there were VGLUT2 endings, those two were also encased with SHANK3. And if you do a pseudocolor image using the staining and combine the signals of VGLUT1 and VGLUT2, we see the co-expression of SHANK3 at essentially every glutamatergic terminal that comes into the granule cell layer. And so with that, we kind of developed this very basic model. So it's not too terribly complex here. We have VGLUT1 and VGLUT2 positive terminals. Both type 1 and type 2 types we found to be consistently encased with these SHANK3 positive dendritic endings. And our hypothesis from this was not based on what we knew about the role of SHANK3 and what we knew about the nature of the synapse, was that SHANK3 is regulating in some way glutamate receptors at the mossy fiber to granule cell synapse. And now we wanted to identify how exactly that was occurring. And so this was a project now primarily led by PhD student Raj Chetri, who I've talked about before. In addition to evaluating animal behavior, he's quite a good electrophysiologist. And so he asked a very simple question using brain slices from both juvenile and adult SHANK3 wild type and knockout mice. And so what he did was he used a compressome VF510 using a sucrose-based slicing protocol and generated some ex vivo live cerebellar slices in the parasagittal orientation. And he performed some voltage clamp recordings from granule cells that again are receiving glutamatergic input from these mossy fibers, and the cells are voltage clamped at minus 60 millivolts. And all recordings were done in the presence of gabazine to block inhibition. And what we're looking at here is a recording of some of those spontaneous EPSCs. And what we see on the left here are recordings from two different granule cells in juvenile wild type mice. And on the right, we have recordings from two different granule cells of juvenile knockout mice. And you see that there is some variability from cell to cell in the frequency of these downward deflections, with each downward deflection here corresponding to an excitatory postsynaptic current. We see there's some variability in the frequency, but for the most part, these EPSCs all tended to be right around 20 picoamps in amplitude, across cells from both genotypes at this juvenile age. And so if you analyze the group data, the average EPSC amplitude was not different between the two groups, between the wild type and the knockout. And the inter-event interval, or the frequency of these events, was also not different between the wild type and the knockout. And then he went on to evaluate EPSCs in adult mice. And this is where we started to see some differences emerge. And just as a reminder, this is also the difference in age between these two groups is where we saw differences emerge in a couple of different behavioral domains that we evaluated. And so again, we have recordings from two different granule cells of adult wild type mice and adult knockout mice. And again, the frequencies were pretty similar between the two groups. But one thing that seemed pretty different almost immediately was that the EPSCs, the amplitude of the postsynaptic currents in the knockout mice were much larger, about 20 to 25% larger or so than those in the wild type mouse. I should say consistently larger. And so if we look at the group data, we did see a significant difference between wild type and knockout EPSC amplitudes with this augmentation or increase in the EPSC amplitude observed in knockout mice. And you can see that the amplitude distribution is indeed shifted to the right towards larger amplitudes. And I would point out that if you look at the amplitude distribution in juvenile mice, there's beginning to be a separation here where that amplitude distribution is shifting to the right. It's just probably not robust enough yet or in enough events to really be significant on the group level in terms of the average. And just as we saw for juvenile mice, there was no significant difference in the frequency. So we wanted to try to understand exactly what was going on here, what was driving this synaptic change, and whether or not AMPA receptors were solely driving this increase in the synaptic EPSC amplitude, or whether NMDA receptors were modified as well. Because remember, SHANK3 has historically been found to be important, at least to some degree, in regulating AMPA, NMDA, and metabotropic glutamate receptors. So the way Raj decided to pursue this question was to try to evaluate glutamate receptor activity in response to a defined and repeatable glutamatergic stimulus that he essentially generated using something called ruby glutamate. So ruby glutamate is a compound here. This is a glutamate attached to essentially a chemical cage that keeps glutamate from acting at and binding to glutamate receptors. And as long as this cage is attached, glutamate is essentially functionally irrelevant. But when this compound is exposed to light, especially short wavelength light like UV light or blue light, that chemical cage breaks, immediately liberates glutamate, and it can activate its receptors. And so Raj set up an experiment where he was able to record inward currents from granule cells in the presence of zero magnesium, such that both AMPA and NMDA receptors were able to respond to glutamate in this configuration. And then he would uncage glutamate only over the granule cell that was being recorded from, and was essentially making his own glutamate receptor, or his only glutamate stimulus that was always the same concentration and lasted for the same duration. So effectively defining the input to the system so that we could very specifically evaluate the postsynaptic response to glutamate. And so what happens when he bathes this tissue in ruby glutamate and then exposes a granule cell to light, and in this case, it's a 50 millisecond long blue light pulse, he sees that there's a rapid inward current. And in this case, that inward current, as you'll see, corresponds to both the current mediated by AMPA receptors and NMDA receptors. And here, gabazine to block GABA A receptors and TTX, tetrodotoxin, to block voltage-gated sodium channels was also present. And then when he repeated these experiments and then added NBQX to block AMPA receptors, he saw that there was a reduction in the response. And this reduction in the presence of NBQX corresponded to the response generated by NMDA receptors, and the subtraction of the two allowed us to evaluate the response mediated specifically by the AMPA receptor. And so when Raj did this experiment for cells from both groups, both wild type and SHANK3 knockout mice, he found that the NMDA receptor response was similar in amplitude between the two groups. But the AMPA response was augmented and larger in the knockout mouse than in the wild type mouse. And it was this AMPA receptor mediated response that was driving this increase in the combined glutamate response observed when both AMPA and NMDA receptors were each contributing to that response. And as he went on to evaluate the glutamatergic response across both groups, he found that indeed there was a significant increase in the response to glutamate when it was mediated both by AMPA and NMDA receptors. But if he evaluated only the AMPA component, that was where we saw a significant increase in the amplitude in SHANK3 knockout mice, whereas the amplitude of the NMDA receptor mediated response was not different between the groups. And we had done some other synaptic stimulation that had suggested the same thing was the case. And then finally, because the AMPA receptor response was increasing and the NMDA receptor response was remaining constant, the AMPA to NMDA receptor ratio was changing. But again, in this case, that was driven by the AMPA augmentation and not NMDA receptor changes. And so the next thing Raj evaluated was some other kinetic properties about the glutamate receptor-driven synaptic response. And he simply just looked at the decay rate of these glutamatergic EPSCs. And what he found was that the EPSC in the SHANK3 knockout mouse was significantly faster than the decay of the EPSC in the wild type mouse. And this was significant across the groups, between the two groups, in that knockout mice did have a much more rapid EPSC decay. Now, at this point, the things that we have noticed about synaptic changes in response to or due to the absence of SHANK3 and cerebellar granule cells is that the AMPA receptor response becomes larger and there is a more rapid AMPA receptor EPSC decay. Now, these two properties are often things that we would think about as being attributed to postsynaptic changes. And we've done some other experiments to evaluate presynaptic function and didn't see any differences between the groups. And so at this point, we're really starting to think that there's something happening on the postsynaptic side in the absence of SHANK3 that's changing the way in which AMPA receptors respond to glutamate. So Raj worked with a medical student who was in the lab the last year, Matt Woodbury, and Matt did some immunohistochemical staining of SHANK3 and the two major AMPA receptor subunits that granule cells express. So he evaluated the expression of SHANK3, GluA4, and GluA2 subunits. And what he found was that sometimes SHANK3 colocalized with GluA4, sometimes SHANK3 colocalized with GluA2, but most often SHANK3 colocalized with both GluA4 and GluA2, indicating that SHANK3 seemed to have, at least anatomically, a pretty strong relationship with the location of GluA2 and GluA4. Now, we're not able to distinguish whether or not these receptors were in the membrane or not, but the likelihood that SHANK3 is regulating that process of whether they're in the membrane or not, based on what we know about the role of SHANK3, seem pretty likely. Now, at this point, if you know anything about the role of the GluA2 subunit, you know that the fact that the GluA2 subunit is present brings the likelihood that there are a number of different functional properties that could be altered specifically in response to the GluA2 subunit. So unlike other AMPA receptor subunits, the GluA2 subunit has the ability to be edited at site 607 and edited in a way that makes the AMPA receptor calcium impermeable, such that any AMPA receptor that is lacking a GluA2 subunit, since most GluA2 subunits are edited, any AMPA receptor lacking that subunit has increased calcium permeability. In addition, as you reduce the level of GluA2 expression in an AMPA receptor, going from higher levels to lower levels, we see that there is an increase in the EPSC amplitude. So with less GluA2, we have a larger EPSC. In addition, absence of GluA2 also leads to a more rapid decay of that EPSC. Now, if you can think back just a couple slides, you know that we have already seen in the absence of SHANK3 larger AMPA receptor responses and more rapid AMPA receptor EPSC decay rates. And the final piece of the puzzle, the thing that functionally distinguishes GluA2 from non-GluA2 containing AMPA receptors, is what's called polyamine-dependent inward rectification. So what that means is in the presence of a polyamine like spermine, which isn't always present in the whole cell recording, but in the presence of a polyamine, receptors that contain a GluA2 subunit have a linear IV or current voltage relationship or IV curve. Whereas those that are lacking a GluA2 subunit have a inwardly rectified IV curve, such that the response at the positive potential is not proportional to the response at the negative potential. And so we evaluated this as well. Roz did some recordings of the EPSC response in granule cells when mossy fibers were electrically stimulated. And in this case, he put a polyamine spermine inside the recording pipette to see if he could identify any changes in this rectification property. And what he saw was, not surprisingly at this point, we had started to have some suspicions that GluA2 levels were changing in the synapse in response to a loss of SHANK3. And so he saw that when you normalize the EPSC response for the amplitude at the negative potential here, and then compare the response at the positive potential, the responses of these two were roughly linear if you were to plot those on a standard IV curve. Whereas we saw a reduction in the response at the positive potential in response to synaptic stimulation in the knockout mouse, indicating that there was some rectification that was going on, rectification in the case of the AMPA receptor mediated response. And the way that we evaluate this rectification is to use what's called a rectification index, which is taking a ratio of the response at +40 to the amplitude of the response at -80. Now, a linear IV curve or linear response here without any rectification would result in a rectification index value of 0.5. Now, what we saw in our wild type mice was that the rectification index value was pretty close to 0.5. There was some variability there, but on average, it was higher or in the 0.5, 0.45 range. Whereas the rectification index value for granule cells from the knockout mouse was more like 0.2, 0.25, indicating that there was some rectification going on. And now with the presence of polyamine-dependent inward rectification, we really have checked off all three of these major functional parameters in the case of our SHANK3 knockout mice to suggest that there is indeed a reduction of GluA2 containing AMPA receptors at those mossy fiber granule cell synapses. And right now, I'll wrap up this part of the talk before I move on to the next. And so in our SHANK3 mice, what we have here on the behavioral side is that a subset of behaviors change specifically with age. And what we noticed was that as animals get older, their motor coordination becomes much worse than it was when they were juveniles. They start to display reduced social preference and increased anxiety-like behavior. And these behaviors specifically paralleled the timing of synaptic changes that we observed at the mossy fiber to granule cell synapse. Specifically, this increase in AMPA receptor mediated responses at that synapse that seems at this point to be due to a reduced integration of calcium impermeable GluA2 containing AMPA receptors. And now what we're working on is trying to understand whether granule cell specific deletion of SHANK3 can reproduce some of these behavioral changes, and if that alone can drive these changes in GluA2 expression. And then also, this isn't everything we're doing in this, but I think our other major question at this point, based on the GluA2 changes, is around what the consequences are. So how does this affect plasticity at this synapse? Does it impact just basal excitability of granule cells? And because we would expect there to be some increased calcium permeability of AMPA receptors in the SHANK3 knockout, is there some potential of excitotoxicity that's occurring here? And so with that, I want to shift gears and talk about another story that my lab has been working on. And this is a project led by a PhD student, Savannah Brannon, where she's trying to explore and understand this interaction between sex and early life stress exposure and how that affects catecholaminergic signaling, specifically activity of neurons in the locus coeruleus, in a way that might alter cognitive and affective behavior. So to begin with, I want to just give a brief note here about adverse childhood experiences. So adverse childhood experiences, also known as ACEs, may be grouped in a number of different categories, from abuse to neglect to household challenges like having a parent or a family member suffering from mental illness, substance abuse, income limitations, and all sorts of things fall into this group of adverse childhood experiences. And many of us have experienced or could have experienced adverse childhood experiences. Oftentimes 0 to 1 is pretty common. But unfortunately, as individuals experience more and more adverse childhood experiences, the quality of their mental health and risk for mental disorders becomes worse. So the risk goes up for mental disorders and the quality of their mental health goes down. So here we're looking at a percentage of individuals based on the number of ACEs that they were exposed to, anywhere from 0 up to greater than 4, and how they rated themselves on a scale. And they indicate that individuals with increased ACE exposure tend to have more persistent feelings of sadness, hopelessness. They also engage in more suicidal thoughts and ideations, as well as attempts. This increased ACE exposure also seems to be linked with other neuropsychiatric disorders or conditions like ADHD and generalized anxiety disorder. And the problem is we don't really have a very good handle on the mechanisms at play when, in terms of what changes in the brain or the neuroadaptations that occur in response to some of these early childhood experiences. We know a lot about what happens in the brain in response to acute stressors, but in terms of early developmentally timed stressors and their persistent impact on brain function is much less well understood. And so we sort of asked the question, taking our approach from mouse models in which we try to identify and understand mechanisms. We simply asked, how would an increased ACE exposure, sort of ACE in quotation marks here, because we're thinking about mice, animal models in this case, but how would increased ACE exposure affect cognitive and affective behavior in a rodent model? Could we recapitulate or reproduce a similar type of behavioral change in an animal model based on their exposure to critically timed early life stress? And so Savannah wanted to develop a model of this, which she's done over the last year or two in the lab. And for her, the criteria for this model were that it have some construct validity, like it should be unpredictable, there should be some deprivation involved, and there should be some perception for the mouse, in this case, that there would be a potential negative outcome. And because critical periods during development are really important for how experiences affect outcomes later, she wanted to target two time periods specifically. So an early and a late time period corresponding to childhood and adolescence. And the protocol she developed looks something like this. So we use bulb C mice from Jackson Labs. And we chose bulb C for this part of the study because this strain already has a sort of genetically encoded, due to it being an inbred strain in this case, a genetically encoded and increased anxiety-like behavioral phenotype. And we were interested in understanding how that predisposition for anxiety-like behavior might interact with exposure to early life variable stress. And so in these mice, a cohort of them was dedicated to being control mice that were simply handled for 5 minutes after weaning. But the mice that underwent early life variable stress, or ELVS, first experienced 4 hours a day of maternal separation from P6 to P16. And then they were weaned a few days early at P17. And then they underwent exposure to a 10-day long unpredictable variable stress. And this was repeated stressors of predator odor, restraint, circadian disruption, forced cold swim, and social isolation. And after that, they were allowed to recover for a week or two before they were evaluated on a few different innate behavioral assays. And then a subset of these mice were used for electrophysiology studies, and others were sent on to be trained in 5-choice serial reaction time tasks in order to evaluate their attention behavior. And so one of the differences that we saw in terms of the behavior of these mice that were exposed to early life variable stress was that regardless of sex, stress-exposed mice displayed increased activity in the open field. So we have a significant hyperlocomotion or an enhanced activity, spontaneous motor activity or exploratory behavior in both male and female mice relative to male and female controls when those mice were exposed to early life variable stress. And I mentioned a subset of these mice were evaluated in 5-choice serial reaction time task. And this is a task where mice are asked to pay attention to and choose a location of a stimulus, a light stimulus that's presented in order to receive a reward. And the typical training procedures and time frame constraints for this behavior are relatively easy for mice to do once they've learned it. So here we have the accuracy, that is the accuracy of a mouse to choose the port, the nose poke that was correct, that was lit in an immediately preceding time period. So if you look at the behavior performance or accuracy of control mice in blue and ELVS mice in orange, you see that on baseline parameters, which are relatively easy, they performed pretty similarly. But if we increase the intertrial interval, now we see a significant reduction in the accuracy of ELVS exposed mice compared to control mice. Now, we didn't see that same disruption when the intertrial interval was shortened. And I'll come back to talking about why we think that is here in a second. And then the third iteration of this testing procedure was done where we reduced the stimulus duration. So rather than a longer flash, the flash of light that an animal had to attend to was much shorter. And in this case, we also saw a reduction in the performance accuracy of ELVS exposed mice. And we think that the reduction in these two time domains probably has to do to some degree with reduced ability to maintain attention within the specific parameters. So with a longer intertrial interval, they would have to have heightened attention maintained for a longer period. And to detect a shorter duration stimulus, they would also have to maintain their level of sustained attention for a longer period. And some people refer to this as vigilance. And so we also just as a follow-up here, we didn't see any change in any change as it relates to each group in terms of the number of trials that were omitted. And classically, these are two of the major parameters that are evaluated in this sort of behavioral task. So regardless of sex, in ELVS exposed mice, what we are seeing at this point is hyperlocomotion and some attention deficits. One of the things I haven't talked about yet, though, is affective behavior or anxiety-like behavior. In addition to evaluating an animal's activity in the open field, we also evaluated their time in the center duration. And by we, of course, I'm primarily talking about work done by Savannah here again. And so when you evaluate the time in the center of an open field, which again is thought to correspond to the level of anxiety-like behavior in animal experiences, we saw specifically in female mice who were exposed to stress a significant reduction in center duration time in the open field, indicating or suggesting an increase in anxiety-like behavior, specifically in females, in response to early life stress. An additional assay of the same type of anxiety-like behavior is often done in an elevated zero maze that has 4 quadrants, with 2 of those quadrants having tall arms that kind of shield the sides, and two other quadrants having open arms. And for similar reasons, we use the time that an animal spends in the open arm as an indicator of their anxiety-like behavior. And again, we saw that specifically in females exposed to early life variable stress, there was a significant reduction in the open arm duration time. And so now together, what we're seeing is regardless of sex, in this model that we've developed is hyperlocomotion and attention deficits, along with a female-specific increase in anxiety-like behavior. Now, as we thought about what might be driving some of these behavioral changes, one of the things that, one of the parts of the brain that immediately came to mind was the locus coeruleus. This is a relatively small part of the brain that sits just below the cerebellum, actually. And these are the neurons in the locus coeruleus, or LC. These are the neurons that are releasing norepinephrine throughout the central nervous system. And they release norepinephrine to shape a number of different behavioral domains and behavioral processes, or processing linked to those behaviors, whether it's salience detection, learning and memory, attention broadly, arousal, or pain sensation, pain evaluation. The locus coeruleus has a number of different functions and roles in terms of how it uses norepinephrine behavior. And so as we were starting to evaluate the effect of locus coeruleus, we started first by evaluating the activity and stress responsivity, and I'll show you what I mean by that here in a second, in a mouse model expressing TD tomato under the control of DBH, or dopamine beta hydroxylase. And so these mice are expressing TD tomato under the control of a locus coeruleus specific promoter. And so we made brain slices, again, using the VF510 comprestome, or these VZ510 comprestome. And in this case, we use an NMDG-based slice preparation in methyl D-glucamine. And if you look at the brain slices that contain the locus coeruleus, you see the DIC image here and the TD tomato expression that corresponds to that DIC image here. And so we did see, specifically in the LC, individual neurons that were expressing TD tomato. And to begin with, Savannah wanted to look at the way in which these LC neurons responded to CRF, or corticotropin releasing factor. This is the peptide or hormone that's released centrally in response to stress, and has been shown before to modulate the activity of neurons in the LC. And what she found was that both in male and female DBH-TD tomato mice, 500 nanomolar CRF would significantly increase the excitability of LC neurons in this brain area. Now, I'm not going to get into it today, but others have also shown that the way in which CRF1 receptors that are mediating this response to CRF, the way in which they signal in the LC in male and female mice might be different between sexes for a number of different reasons. And because of that, we asked this question: is sex-specific variation in EOVS-induced behavior something that most likely would be engaging a release of CRF that would act on the LC at early time points? Might this sex-specific variation in EOVS-induced behavior correlate with sex-specific changes in LC physiology? And so to begin with, Savannah recorded from LC neurons in these bulb C mice that we had evaluated the behavior of after stress. And she found that both males and females, sorry, these are evoked responses in response to 80 picoamps of positive current injection and 30 picoamps of hyperpolarizing current injection. She found that the evoked response, or evoked electrical excitability of these LC neurons, was reduced in response to ELVS for both male and female mice relative to their male and female control counterparts. And if you evaluate the excitability across a range of current injections, we consistently saw a reduction in the evoked excitability of ELVS-exposed male and ELVS-exposed female mice. And she went on to also look at the spontaneous excitability of these LC neurons, and not surprisingly here, and as a nice parallel to evaluate the evoked excitability, again, ELVS exposure in male and female mice caused a reduction in the excitability of these LC neurons relative to control male and control female mice. And if you look at the group data here, you can see that there was a reduction in the spontaneous firing rate in the case of male and female EOVS exposed mice, again, compared to control male and control female mice. Now, as we tried to think about what might be driving this reduced excitability, we evaluated the resting membrane potential. And what Savannah noticed was that in the males, things seem to make sense here. The LC neurons from male mice were significantly hyperpolarized just at a resting state relative to the resting potential of control male mice. But surprisingly, we did not see that same change in the resting potential in female mice. In fact, we saw the opposite. So there seemed to be something, two different mechanisms possibly that will be contributing to this change in excitability in response to ELVS that was sex dependent. So the way that Savannah tried to address this was to repeat these recordings, but this time, instead of letting neurons sit at their natural resting potential, she injected a little bit of current at steady state to maintain their resting potential at about minus 60 millivolts, so that all neurons were starting at the same resting potential. Now, she still did see a reduction in the evoked excitability of neurons from ELVS male mice, LC neurons. And then in female mice, she also saw a reduction, but there was something now that had emerged that we hadn't seen before, very specific to what was happening in ELVS exposed female mice. And that was that at these hyperpolarized potentials, modestly hyperpolarized potentials, we were seeing an exceptionally long delay time until the first action potential was initiated. So again, the group as a whole, ELVS male and female mice elicited or demonstrated reduced excitability here and here. But as we evaluated the action potential delay time, only ELVS female mice were displaying a significant and rather large enhancement of this time to delay to the very first action potential in response to current injection in these recordings. And so with that, I want to summarize everything, including the behavior, but also some of the specific aspects of the electrophysiology that I just mentioned. And so behaviorally, in our model of early life variable stress exposure using bulb C mice, we're seeing hyperlocomotion and sustained, reduced ability to sustain attention or reduced vigilance in both male and female mice. We also see an increase in anxiety-like behavior only in females. And corresponding to that, if we look at the electrophysiological changes in neurons in the locus coeruleus, that there's a lot of support for them being involved in shaping these types of behaviors. We see that in both sexes, there is a reduction in the excitability of LC neurons at a few weeks after exposure to, or the last period of exposure within the ELVS procedure. But specifically in females, there was this unique increase in the action potential delay. And whether or not and how these two specific types of electrophysiological changes correspond to these behavioral changes, that's something that we're actively evaluating right now, in addition to trying to understand how other rodent strains, maybe those like the C57 that do not have an innate elevation in anxiety-like behavior, how do they respond or change their behavior when exposed to early life variable stress? And so with that, I will just like to thank everybody who's contributed to this work. So on the right here, we have our former lab members. Those in bold have contributed in some way, largely behavioral assessment of mice in different configurations, but they've contributed in some way to the work that I talked about today. We have a few medical students in the lab, Lauren and Lupe, but the team primarily right now are these 5 individuals. I talked a lot about the work done by Raj and Savannah. James has just recently moved on to be a medical student here at SIU, but he contributed a lot as well. I didn't get a chance to talk about some of the circuit and track tracing work involving the cerebellum and other structures that postdoc Timmy is doing. And of course, we have our lab manager, Hunter, who helps us keep organized and make sure that the mouse colony stays on track and so on. And lastly, probably just as important, I'd like to thank those that are sponsoring and funding this work. We have a grant from both NIMH, National Institute of Mental Health, and IDCD as well. And so with that, I'll finish up, and I'd like to open up to any questions if there are any. And if there are not today, everybody who might view this either synchronously or asynchronously is welcome to email me, or I think email anybody at Precisionary, and Chloe's probably going to say something about that. If you have any questions about our slice preparations and or the data that I talked about today, I'd be more than happy to talk with you about it.
Great. Thank you so much for your presentation, Dr. Richardson. So yes, like previously mentioned, if anybody does have any questions after viewing this, please reach out to us or Dr. Richardson personally. So with that being said, I will stop the recording and thank you so much.
Thank you.